Okay, can we go back to the start? Bob, I must say I'm very impressed by this meeting already. It's amazing how many people have interest in XP and have contributed tremendously to our increase in understanding. My interest in XP really begins back in when I first came to the United States in 1964. This was essentially to be a postdoc for a few years and intending to return to the United Kingdom, but that, as you can tell, never happened. However, it also was the beginning of several unfortunate personal experiences. Within a couple of years of being in Boston, the PI who was funding my postdoc decided to leave for New Zealand. So I was abandoned completely on my own with no money and no support. But I was very lucky to meet Robert Painter, and he was setting up a new lab, which was in San Francisco. So next slide, please. And he was setting up the Laboratory of Radiobiology. And this lab had a long run for 30 years, supported by the Department of Energy. The director next is Harvey Patt. He was well known for sulfhydryls and radiation biology of the bone marrow. Next, his second in command, Sheldon Wolf, had set up, had a career in chromosome aberrations from radiation. He regretted for many years that he never actually used the word repair because he felt he was left behind. However, that's old history. Then the third faculty member, Robert Painter, was my immediate supervisor. He was, at that time, became very well known for actually making tritiated thymidine. This became a workhorse in studying cell biology for many years. And the next, please. He showed that you could label cells with tritiated thymidine using radioautographs, detecting S-phase cells with dense labeling. But what was a surprise was in 1964, Bob Painter, with his support by Ron Rasmussen, they discovered and reported that not only could you label S-phase cells, but if you had irradiated the cells first with UV, all the cells would be labeled at a lower level. And we spent many, many happy, shall I say, happy days counting these things as a way to quantify the effect of ultraviolet light. But at the same time, we weren't really sure quite what it represented. It could have been a completely artificial stimulation of replication. So there was considerable argument and competition as to whether it had any biological significance. The major step came, and well, I was tasked with the job of trying to make mutations in human cells in culture and see if we could use that route to identify the function of unscheduled DNA synthesis. That wasn't a success, but what was, in the next slide, I can see what was a surprise was that at a San Francisco medical meeting, there was a report which was summarized by David Perlman, who was an outstanding science correspondent in San Francisco. This was a family pattern in cancer. It was a family with 6 or 7 children. They all had skin cancer. It seemed to be associated with the sun. And in Perlman's comment, they thought that this represented a familial cause that could be traced to sun exposure. So both a genetic and an environmental impact. Now, I came across this because in a quiet moment with nothing else to do in the lab, I was just reading the San Francisco Chronicle. And I don't recommend this as a policy to anyone who's directing students, but don't get in their way. In fact, when I thought this represented perhaps a mutation that was relevant to sunlight and cancer, I came to Bob with this suggestion. And the next slide. He was a very impressive man with a very dry sense of humor. And his remark was, next slide, please. His remark to me was, a crazy idea, but at your stage, you've nothing to lose. Now, there's some professors and mentors who like to encourage, and there's others who like to challenge. Bob was one who would challenge. And so the next slide, please. And so on that suggestion that I gave to him that XP might be a human mutation in DNA repair, it was incredibly fortunate of combination of factors that led to this. Next, please. So within a short time, with the support of members of the dermatology department of UCSF, they identified as a number of California patients who had xeroderma pigmentosum. And one was an adult male with skin cancer, but no other complications. Two others were children, SIBs, that had melanoma, had skin cancer and leukemia. And so this was interesting that these three patients already showed to us that there were substantial differences from patient to patient and the possibility of additional disorders, not only skin, but leukemia and possibly others. Now, at that time, it is difficult to think back to understand how little we had in the way of tools for genetic analysis. There was no polymerase change reaction. There was no cell cloning. There was no protein or in vitro assays. But it was cell biology. And several groups established cell fusion experiments by fusing cells from different patients and determining whether they showed normal unscheduled DNA synthesis or no. They could be classified in complementation groups. So within a short time, we could classify xeroderma pigmentosum into 7 groups, A, B, C, D, E, F, G. And this was the way the field stood for quite some time. There was several years of an analysis of the biology of the complementation groups. Veronica Maher and Shazzy McCormick showed that these were all deficient in repair that resulted in increased UV mutagenesis in vitro, which was in fact technically very difficult, and very few other people have achieved what they achieved. Now, we decided that what we would like to do would feed back some support and help to patients. So we set up an informal off-record testing service in which we invited dermatologists to donate biopsies from their patients, and we would establish in culture, measure DNA repair and UV sensitivity, and return a potential diagnosis. This was well before informed consent or government regulations came into force, and we were able to do this for at least a decade. Not only patient diagnosis, but we did some substantial prenatal diagnosis as well, and showing that it was all feasible as a diagnostic process. Now, in the process, we found there was a considerable number of ultraviolet sensitive patients whose cells were given to us and yet we could not find any DNA repair deficiency. So there was obviously other sources for UV sensitivity, and these we have stored, but have yet to be analyzed. So next, please. For about 20 years, the major progress was in the biological analysis of cells from XP patients, but no molecular analysis. But the most significant approach happened in 1988 when Rick Wood developed the in vitro method for detecting and dissecting the components of DNA repair. At that time, soon after, there was quite a surprise to many of us, in which Schaefer, Higley, Hohemakers, and others demonstrated that nucleotide excision repair was strongly linked to transcription. And in fact, one of the major transcription factors that they had analyzed contained a multi-protein complex in which there were two helicases that were actually XP genes. It's worth pointing out that from the discovery of XP in 1968, it took until 1990 for the first gene to be cloned and sequenced. So it was a long and tough progress. In addition to this, next slide. Just move to the next, please, Bob. Yeah. So these two components were critical for the unwinding of DNA during in vitro repair. Next, please. And after that, during this period, as we were looking and performing our patient service, we found patients who were obviously clinically diagnosed as XP, but they did not have repair deficiency. And this was an embarrassment. We weren't quite sure what to do with these. And I felt that the important thing was to trust the clinical diagnosis. So we decided to name them as variants and publish this and next slide, please. Publish this next in 1972, in which we described variants with normal DNA repair and normal sensitivity to ultraviolet light. These have become very interesting and important group of patients. It was, for a long time, studies done in cell biology showing that these had replication defects and they were sensitive to inhibitors such as caffeine, but the precise cause was difficult to know. So we hedged our bets, named them as variants, and moved on. And it was not till many years later, in the 1990s, that Johnson and Prakash, our group, showed that this was a mutation in a low-fidelity DNA polymerase. And this harked back to mutagenesis study that Veronica Maher and McCormack picked up on, and they studied the XP variants and showed that it did have a very high UV-induced mutation rate, so that the relationship for UV-induced mutagenesis and skin cancer was retained both in excision repair and in the polymerase deficiency. Next, please. This polymerase was found to be a member of a large family of polymerases that are involved in replication and repair and bypass of photoproducts. The polyether, which was the one associated with the XV variant, was the most error-prone, with an error of 1%, compared to pol alpha with a very low, which is the replication polymerase. And its structure showed a very interesting feature of an active site that would accommodate a photoproduct and allow in wild-type version replicate the past photoproducts. In its absence, mutagenesis would be very, very elevated. Next, please. Now, it could be thought that at this stage, there was nothing new, nothing surprising. But I want to end referring to this particular patient. Mark, Muriel and Clark Lambert, are friends of mine in New Jersey, had a unique XP patient whose squamous carcinomas spontaneously regressed. He had melanomas which needed to be surgically removed, but not the cutaneous squamous carcinomas. So it was a considerable puzzle. We confirmed its diagnosis with XPC using an antibody and immunohistochemistry. But they sent the tumor samples to us, and we carried out an exome sequencing analysis of the tumor and found that not only did we have an XPC mutation, a homozygosity, but in the tumor, it was almost also homozygous for a mutation in a protein, ceramide kinase-like protein. And there are mutations already known. Homozygous mutations would cause retinitis pigmentosum. Now, this is not a disease that this patient had. So we were faced with a question, exactly what is functioning here? What is the cause of the regression? And it was already known, and which we will show you in a moment, the loss of function of this protein made tumors reactive oxygen sensitive. Next, please. In fact, in squamous carcinomas, this protein circle is highly expressed compared to normal human keratinocytes. Next, please. So using siRNA and hydrogen peroxide as an oxidative challenge, we showed that in a siRNA against circle would increase levels of apoptosis considerably high compared to normal cells and compared to a scrambled RNA. This is where we stand at the moment. The analysis of the function of circle we published in British Journal of Dermatology early this year. Muriel and Clark Lambert very generously sent me biopsies, which we sent into culture. And these are still under investigation. Next slide, please. We're dealing with a situation that is potentially a unique but very interesting phenomenon. So the question we need to ask is, do the patients carry a pre-existing circle mutation? It is unlikely that the person is carrying these as a homozygous mutation because there's no evidence for retinitis pigmentosum. So presumably he's heterozygous. So the question is, could the circle mutation be generated by mutagenesis in the XP cells during tumor development so that a novel circle mutation is added to a pre-existing one to make the tumor homozygous? This is the working hypothesis. And at the moment, all I can say is it's subject to patient access. Like so many experiences in clinical research, since there's very little that we can actually do for this patient, it's very difficult to have access for continued analysis. So work is carrying on at the moment in my lab to ask whether we can get answers to these questions. So let me, next slide, please. So perhaps, just as an aside, we've already filed a patent for Circle if it causes spontaneous regression of a tumor. It has potential for therapeutic uses. So it might conceivably be a competition to the Estee Lauder night repair that I might say is actually not a DNA repair protein. Next, please. But it remains, as I'm wrapping up, that a well-protected patient, courtesy of NIH, Ken Kramer's daughter's sketch, protection is the best of all. And this is a patient, an XPA patient, who's allowed me to use her photograph. I diagnosed her when she was less than a year old. She's in her 30s or 40s now, an XPA patient, but with careful protection. She has very, very little skin symptoms, showing an extremely beneficial effect of protection. Now, my final comment, personal, is that I come from a university campus that's well known for biotechnology development. And so I can't be one without a biotechnology component. So the next, please. I can see the next. There is already out in the field, Cleaver's repair. Unfortunately, it's not DNA repair, it's automobile, but someone already has stolen the march on me. Next, please. So in conclusion, I would say I'm very grateful for the colleagues, especially those who've helped me the last 10 or 20 years when our lab has essentially been closed for the virus, and also because funding was abandoned. And I would also be very grateful for patients who were willingly in hope participated in our research, and many that I know in some California area. I just wish we had more to give than what we said in 1966. Protection is better than treatment, so keep out of the sun. Thank you.
That's a really wonderful talk. Thank you so much, Jim. I'll just change a few buttons around. That was absolutely fascinating, and I think the circle stuff has really was really among the surprises that followed. I just wonder whether the colleagues of ours who are here, some of whom may remember the early days as well, and I just wonder, we'll open this up to discussion, but I was just wondering whether Ken Kramer or Alan Lehman or Chicago or Alain or any of the other colleagues around here who may have memories of the earlier days when XP research was in its infancy, have any comments or questions to ask to Jim.
Right, I can make a comment, I guess. So I did my PhD from 1967 to 1970 at the Institute of Cancer Research at Sutton, and I was working on DNA repair, which was at the time regarded as a very obscure and esoteric topic. And I think it was about in the second year of my PhD in 1968, someone came to give a seminar, a Brit from California, name of James Cleaver, and he told us this amazing story of XP, which really was the start of it blossoming from an obscure and esoteric subject to one which actually had a human connection, which we know now has got not only with XP, but the many disorders and the central place of DNA repair in carcinogenesis.
Thank you very much.
This is Alain. Okay, Jim, very nice talk. In one of the first slides you showed, you described 2 XPA patients, which were the first XPA patients that you studied. And you said that they had leukemia at that time. This is true, the 2 XPA had leukemia?
Yeah, they were, we confirmed they were XPA later. And I have no idea what's happened after that. All we obtained was a clinical diagnosis and some skin biopsies. I never actually met the patients in person. And so there's very little else I can say. It's one of the interesting examples that other diseases are associated with XP, sometimes in more mature patients. But there's this surprising one that we've just discovered that has a circle association that actually is anti-carcinogenesis.
Okay, no, it's because more and more we have XP patients, not XPA, but XPC patients who develop myelodysplasia followed by leukemia 20 and 25, something like that. Yeah. And this is really a very, very high frequency, at least in the patient of XPC that we have in France coming from North Africa.
Mm-hmm.
Yes, just I'd like to add a little, just like Alan said, Dr. Cleaver actually came to NIH and gave a talk before it was published, and Dr. J. Robbins heard that talk, and I started working in his lab. And he said, well, we better follow up on this. And then we started growing Sendai virus in eggs to do cell fusion studies. And we did did the fusions, and we came up with 3 complementation groups. And then Dr. Boatsman group in the Netherlands had previously found 2 complementation groups. We all got together and exchanged our cells very nicely. There was a nice meeting in the Netherlands, and I brought my cells and brought them back there. Jan knows about this. And then we fused back and forth, and we came up with 5 complementation groups. We named them in order of increasing levels of repair. A was the lowest, and E was the highest. And then the 3 patients that Dr. Robbins found all had reduced repair, but the 4th one had normal repair and clear XP. And that was the first variant. And we all were concerned and happy that Dr. Cleaver didn't start out with that 4th patient, XP variant, and it probably would have given up at that point rather than seeing the others with reduced repair. So, thank you. Nice presentation.